Hi, my name is Jędrzej Świeżewski. I'm the head of AI at Epsilon, and I'd like to present to you a talk that I gave at BioTechX in Basel last month. The title is The Future of Pharma, Leveraging AI for Quick Detection of Protein Crystals. What we will be talking about is I'll try to argue that artificial intelligence can be really powerful when it is applied right. And in fact, I won't be talking about any general artificial intelligence model, but rather I'll focus on so-called specialized AI, or simply machine learning models that can be shown to be really powerful when applied right. So what do I mean by being applied right? I'll focus on three main areas. One is that the cutting edge tools and methods need to be leveraged. Secondly, the whole process, the pipeline, the project needs to be reproducible. And lastly, I'd like to show that we don't need to be limited by how humans do a certain task. Sometimes the machine learning models can go beyond what is possible for humans. And I'll illustrate each of those 3 points with a specific case study from our past projects. First, showing you how we've beaten the state-of-the-art model in the area of protein crystallization imagery. Second, I'll present a case study of a collaboration with AstraZeneca. And lastly, I will talk about a collaboration we're kicking off with a top crystallization center. There will be also a bonus slide at the end of the talk, so stay tuned. Now, in the main part of the presentation, I will talk about protein crystallography. It is a major tool that is used in drug discovery still, and it has been for the past few decades. And in a nutshell, it boils down to a process in which a protein is prepared, and then it is crystallized in a so-called crystallization screen, where many different conditions are proposed, such that those in which the protein crystallizes can be found. And once this is done, the protein crystal is extracted to then undergo X-ray crystallography imaging, which basically allows the scientists to understand the structure of that protein, which can lead to understanding how to bind to it and can eventually lead to a new drug being developed. Now, we will focus at the mundane part of this process, which is crystallization screening. In that process, multiple conditions are being proposed, as I said. Think of different pH levels, maybe different temperatures, different solutions, in which the given protein is tested, whether it crystallizes in them. And basically images like the ones I've shown you on this slide are generated by the robots typically performing this crystallization screening. And in those images, you see droplets in individual wells. And then the scientists need to find those droplets or those images among thousands in which crystals actually did form. Now, it turns out that although in this pair of images it seems rather obvious where the crystallization happened, it actually is not that obvious, typically. And in fact, when studied systematically, it turns out that crystallographers agree with each other whether a certain image presents crystallization results or not, only 70 to 90% of the time, depending on the exact study. Now, even more strikingly, when in a recent study, scientists presented the same images, it was actually 1,000 images that they presented to a professional crystallographer to assess what was the crystallization outcome. Was it the crystal? Is it just phase separation or precipitation? Or maybe there was nothing. And then they presented the same crystallographer with the same set of images after 6 months, and they measured the self-agreement rate, and it was just 83%. So this shows that while in some images it seems that it's maybe clear what the distinction is, in many cases this is far from obvious. And in fact, I've played a tiny trick on you, because showing those two images, I've sort of suggested that one of them presents crystals and the other one doesn't, where in fact crystals are present in both of them. And in particular, the one on the right, the crystal is where the green circle shows up. And in fact, I have to say that this is a real image from one of our datasets that we've studied, and our machine learning trained model has confidently identified that crystal as well. All right, so this is the intro. Now let's look at the state of the art. Is it a problem that actually can be solved or aided with machine learning? And yes, the answer is positive. There is a collaboration called MARCO that has been launched several years ago by several leading institutes and companies in the field. And after forming this collaboration, they have gathered a large data set, trained a machine learning model, and actually open sourced it. It's commonly available, and in fact, it's very popular. A lot of crystallization teams have tested this model. And the problem is that this model doesn't really generalize that well to new data sets. And in fact, when we have looked at this model, we were asking ourselves, well, can we improve on it? And first of all, to benchmark ourselves fairly, we've tried to use the same training set that Marco used and use the same test set so that we have a fair comparison. And just by improving the way the machine learning modeling was done, we've been able to improve on a crucial metric, and that metric is the recall on crystals. So the measure of how many crystals have been actually captured. And importantly, we have not introduced more false positives. So keeping the precision at the same level that the Marco model has, we've improved the recall such that the rate of the missed crystals was reduced by almost a third. Now, the question is, how does this model, the Marco model, perform on your data in a given crystallization setup, not necessarily coming from the teams who contributed the data set to the training data? How does the model generalize? And this is a known problem in the field. A lot of crystallographers that I've spoken with told me that yes, they have tested Marco, but it doesn't work as well on their data. So what we did there is we have picked a certain challenging data set and measured the performance of Marco on it. And you see the performance marked with the red line. It's just 76% accuracy. And then we asked ourselves, okay, so can we find a model and an architecture of a neural network that would allow us in a certain setup to feed it just a bit of the hard images, so from this peculiar data set, from a lab external to the training data. And what is the limit? Like, what is the fewest number of images that we can take to improve the model, and how much of this improvement can we see? And in fact, the results are marked here with the blue line, and you can see that as few as 60 images of crystals plus 60 images of non-crystals, so in total 120 images, already gave us a boost of performance of our model as opposed to Marco by almost 10 percentage points. And as we went into larger data sets, still fairly manageable for crystallization screen standards, if we take 1,000, maybe 2,000 images of crystals, we already see the accuracy boost to the levels of the 90s from the 76 offered by the off-the-shelf Marco model. So the lesson from this slide is that with latest techniques, when the machine learning model is applied right, we can beat the state of the art noticeably. All right, so let's see it in action in a specific case. Now we will move to a case study from a collaboration with AstraZeneca. The way it all started is that the team at AstraZeneca also noticed that Marco is not good enough for them. Therefore, they developed an internal model in a collaboration with University of York. And the model seemed good. On the other hand, the users, the crystallographers, were not that sure that it actually performs confidently. And only after we joined forces with the AstraZeneca teams, we have identified certain shortcomings in how this model was prepared, and more importantly, how this model was assessed. We've improved the robustness of the assessment of the performance of the model, applied also certain technical improvements in how the machine learning modeling was performed. And what we achieved is that when measured on AstraZeneca's production data, the previous model has missed 15% of the crystals, while the model we have proposed misses less than 3% of the crystals, which is again a key performance metric of the machine learning models used for crystallization screens. All right, so we see that with new methods, with robust pipelines and reproducible methodologies, we can actually improve the models. We can achieve really good results. So the question is, can we go beyond what is possible for a human? And here I'd like to just glimpse a certain solution that we are preparing in a collaboration with a top crystallization center. Basically, all the models that we have seen so far used in the field, they focus on so-called bright field imagery. So a certain image type that is collected in crystallization screens. But in fact, typically, more is collected. Also UV light is collected, and then the time sequence, the time-lapse dimension is also collected. So there are multiple images of those wells taken typically in a few days period or in order of few days. And we are preparing a model that will actually leverage all that information to improve even further and possibly be able to show performance of detection of crystals beyond what is possible to humans. Stay tuned for those results. I'll make sure to share them with you as soon as they're available. Now, also an important note is that if you would like to join this collaboration, if you are having also collection of UV light and maybe the time-lapse dimension, and you're thinking of leveraging it in your pipeline, but you're not sure how to best make use of it, reach out because we might help you, and it would be great to talk about a common project. All right. With that said, the summary of my talk is that I hope to have shown you that machine learning can be really powerful. For example, in high content screening or high throughput screens, when it is applied right. There is one more extra point that I would like to make, and that is that it's not enough to have a very powerful model. It's also important to make it accessible, to make it useful to the eventual users. In the case I've presented today, the crystallographers, but maybe these are the different scientists looking at their own screens. And in fact, this is an example that I want to share with you as a closing slide. In particular, when cell painting or high throughput cell imaging assays are used, multiple images of cells, typically live human cells, are collected with certain fluorescence added to them, certain paints added to them. So with different lights, therefore many channels. And once this is collected, the morphology of those cells can be crossed with the genetic understanding of the preparation of this assay to reveal new information. And in fact, we have been involved in 2 such projects recently in a collaboration with the University of Bonn, and you see the list of collaborators on this slide. And I'm proud to say that one of those projects has recently been accepted in Nature Biotechnology and should appear very soon in the journal. So the details will be available. But for now, I want to highlight that here we have not only built machine learning models that revealed new information, but we've also built an interface, a demo of which you can actually see in this slide, which allows the user to quickly browse through the data to understand, slice and dice it, which basically reveals the hidden information that the model has already captured, but it's very hard to uncover otherwise. And in fact, I'm proud to say that the main project we're working on in this respect is still being prepared, and the one that got accepted was just an application of the methods we've developed to a new data set. And it turned out to be extremely useful. So again, one thing is cutting edge, reproducible machine learning models. The other thing is being able to predict, to embed them, to make them accessible to the user. Thank you for your attention, and do reach out if you'd like to chat about a potential project. Thank you.